I would now like to talk about inductive interactions during embryonic development in amphibians. It turns out that a number of the early discoveries in amphibian embryos have now been confirmed as standard mechanisms for development in other vertebrates, including chick mammals and fish. The classical work of Hans Spemann and his student Hilda Mangold using nude embryos pushed the field of developmental biology forward very significantly in the early 1900s. If we look here at Figure 1, this is one classic experiment that really began Spamann's research program, and it was actually an experiment that we had talked about in one of our earlier lectures, earlier in the course, in which he demonstrated that when a fertilized egg was constricted by a ligature, with the nucleus excluded from half the egg, that half remained undivided. At the 16-cell stage, he slightly loosened the ligature, and one nucleus then was allowed to move into that half of the egg that did not have nuclei, and what he found was that that would start dividing. He constricted the ligature further, dividing these into two separate individual eggs that were dividing, and what he ended up with were two completely normal salamander embryos. And of course they were smaller because they started with smaller mass to begin with. Spemann concluded from this experiment that early amphibian nuclei were genetically identical and that each cell was capable of giving rise to an entire organism. However, when Spemann performed a similar experiment with the constriction still longitudinal but perpendicular to the plane of the first cleavage, that is, separating the dorsal from the ventral aspect of the egg, what he found was that one of the pieces of tissue did not really form a normal embryo at all. It formed some tissue, but it didn't form tissues that included neural tissue, brain tissue, and some of the aspects of the anterior part of the embryo. What he noted was that the part that had the gray crescent allowed normal development to take place. However, if the gray crescent was absent, normal development would not take place. And what did this suggest then? Well, it suggested to Spemann at the time that there was something different about the gray crescent. In other words, the egg was not totally uniform in its substance, and there was something in the region of the gray crescent that must be different than other parts of the cytoplasm. And as it turned out then, in future studies, it was found that indeed the gray crescent was important because what that gray crescent did was to give rise to the dorsal lip of the blastopore. And later on it was found that the dorsal lip of the blastopore was important in allowing gastrulation to take place and thus in allowing the embryo to form normally. This shows an example of the gray crescent. We saw this previously, but here you can see a fertilized egg. The gray crescent is right in this area, and this is the area then that is going to form the dorsal lip of the blastopore. It's going to start gastrulation here. This is going to form the primary organizer. Here's the first cleavage division, and ordinarily, in normal development, that first cleavage division will go through the gray crescent area, thus allowing gray crescent to be on both sides of that developing zygote, so that the entire zygote will be involved in the gastrulation process. If you split them at this point, you will get two normal embryos because that gray crescent is there, and hence each will form a dorsal lip of the blastopore. And just to remind you what the dorsal lip of the blastopore looks like, here is a Xenopus laevis embryo. The dorsal lip area, here's the animal hemisphere of the zygote of the embryo. Here we have the vegetal pole. Here is the dorsal lip of the blastopore right here. Involution is taking place, and this is where gastrulation begins. Spemann speculated that the importance of the gray crescent material lies in its ability to initiate gastrulation. In later studies, he performed experiments that showed both statements to be true. He found that the cells of the early gastrula were uncommitted, but that the fates of the late gastrula were determined. This involved exchanging tissues between the gastrulae of two species of newts which had different pigmentations. And you can see that in the experiment at the early gastrulus stages, he took an area of presumptive neural ectoderm, transplanted it to an area which ordinarily would form epidermis, and at the early stage, that presumptive neural ectoderm indeed formed into epidermis. If, however, this was done at a later gastrulus stage, this is the late gastrulus stage now, here's the archenteron that's well formed, here's the archenteron that's just beginning to form, but here now you have presumptive neural ectoderm, you put that in the area of presumptive epidermis, and indeed, even though it ends up on the belly part of the embryo, neural plate tissue forms. What this said was, at this later stage, this development was autonomous. It took place because this had already been determined. So in the first case, the cells of the early newt gastrula exhibited conditional differentiation. In other words, they were induction dependent and would be induced by the surrounding tissues here. And again, as I mentioned, at the later stage now, this tissue has already been induced and committed to form into neural ectoderm. It has been determined. And once that happens, its fate is going to be neural plate no matter where you put it. And so this was a very important discovery that Spemann and his colleagues made, that prospective fate was determined after it had been in place for a given amount of time and would differentiate independent of where it was placed in the embryo. Just to kind of review this information, here we see at the early gastrulus stage, we take prospective neurons or epidermis, put the neurons in the epidermal area, and you would get conditional development because it would form prospective epidermis. It would form epidermis. Took the prospective epidermis, put it with the host region where the neuron would form, it would form neurons. So it's conditional on where you've put it during early development. Late gastrula, the prospective neurons into prospective epidermal area, you still get neurons because it has been committed, it has been induced, and it has been programmed basically for autonomous development. Epidermis the same. If you put it with the neurons, it will still form epidermis because of this autonomous development. I want to now talk about a very classic set of experiments that were done to show what was termed primary induction. In other words, what was the proof that the dorsal lip of the blastopore was the primary organizer that could organize the formation of an entire embryo. And these were some very spectacular experiments that were done by Hans Spemann and his doctoral student Hilda Mangold back in 1924. And what they found was that if they took, and they again used the color-coded embryos, and you could then pick out the tissue that you had transplanted. But what they did then was to take the dorsal lip of the blastopore from this one species, transplanted it onto presumptive epidermis at an early gastrulus stage, and what they found was in that transplanted area, you got a secondary invagination. You got an archenteron forming, not only the original one from the dorsal lip of the blastopore, but now you basically had a new dorsal lip of a blastopore that had formed the archenteron, and it would end up continuing to develop, forming neural tubes, somites, notochords, all of the tissues associated with another embryo. And it had come from the dorsal lip of the blastopore of another species, actually. And what happened then is, as the embryos developed, you ended up with two embryos formed, one from the original dorsal lip of the blastopore and one from a secondary lip. And they also did a series of experiments to show that if you did this at later stages of development, you would get different regions of that dorsal lip, and you would get different areas that would be formed in that secondary embryo, different regions. But if you took it very early, you would get a whole embryo formed. For this work, Spemann and Mangold were awarded the Nobel Prize in Medicine or Physiology, and this was really the first instance where a graduate student with her thesis had won a Nobel Prize, and perhaps it's at this point the only instance. But in any event, it was surrounded by tragedy because Hilda Mangold, living in northern Germany, and it was during the wintertime, was putting gasoline in her stove, and that's the way they heated their apartments in northern Germany at that time. It exploded and led to her tragic death, just prior to the time when she was to have her paper published and when she was to receive the Nobel Prize. And so, although she had a very short career, a terrible end to her career, she contributed so much to science by doing this really seminal work that really changed the field of developmental biology. Since that time, of course, many years have passed, and it was not really until the last 10 or 15 years that further progress has been made on what this primary inducer is. What are the things that make the dorsal lip of the blastopore capable of forming a whole new animal? And it's been found that this is not just true in salamanders, it's true in all of the vertebrates, that there is a dorsal organizing material that's really a primary organizer. So for many years, the mechanisms by how this happened were mysteries. And really, as any great experiment does, it creates more questions than it answered, and certainly that was the case with the work that was done by Spemann and Mangold. It really was the starting point. This primary organizer discovery was the starting point of one of the first truly international scientific collaborations to try to determine what this magic material was that could take tissue that ordinarily wouldn't form much and form it into a living individual that would grow into an adult that could reproduce again. Like I say, it wasn't until the last 10 or 15 years that significant has continued now in this field. The proteins responsible for induction were present in concentrations really in the early stages to be able to analyze them, and plus in the amphibian system, which is where you could really show this and where it had been shown originally, there was too much yolk and lipid material to really get at purifying these small amounts of proteins that were present in that dorsal lip of the blastopore. So with the new recombinant DNA technologies, however, and some of the molecular biology studies where you could look at details of messenger RNA and make clones of these and see what these specific clones would do, it wasn't until this technology developed that further progress has continued to be made. Certainly we are now able to take up these questions that were posed in the 1930s. In turn, many of them are still, however, in the process of being answered unequivocally and mechanistically. At this point, molecular developmental biologists have made significant progress in describing the presence of various paracrine factors, growth factors, specialized molecular receptors, etc., associated with this primary organizer. However, the inductive phenomena that direct development of a new individual organism still remains a mystery as to the exact mechanisms of how this happens. The unequivocal mechanisms of many of the developmental events, then, still are in the process of being elucidated.